The overall goal of this experimental protocol is to measure changes in blood oxygen level dependent activity induced during transcranial electrical stimulation.
Combining transcranial alternating current stimulation with functional magnetic resonance imaging has the potential to answer key questions in cognitive neuroscience. The main advantage of combining these techniques is the possibility to measure brain function while manipulating neural oscillations in a frequency-specific manner.
The implications of this technique extend towards diagnosis and therapy of neurological disorders associated with abnormal synchronization.
Though this technique can provide insight into human visual cognition, it can also be applied to other systems such as motor behavior and memory.
Generally, individuals new to this method might struggle with the complexity of the setup that is required to ensure noise-free and trigger-synchronized experiments.
We became interested in tACS because we saw it as a way of studying the causal wall of oscillations to conscious perception.
Our MRI technician is Ilona Fallet, with Paula Wunsch as our subject volunteer.
Pre-screen all subjects for any contraindications to MR scanning as well as to transcranial alternating current stimulation. When the subject arrives, describe the details of the experiment and obtain informed consent. Begin by using a tape measure to determine the distance on the subject's head from the nasion to the inion, and then from ear to ear over the top of the head.